All right, respiration, cellular respiration. All right, this gets a little thick, a little deep, so reach down, tie your shoelaces, hold on. So really, all of the energy on our planet comes from the sun. Most all of it comes from the sun. Plants take this solar energy, this electromagnetic radiation, they'll bring it in, and through a very complex process, take inorganic carbon dioxide, the energy, fix it, and turn it into stored energy, known as phosphoglyceraldehyde. Modern books now call it a glyceraldehyde 3-phosphate. And then the plant will take that and turn it into everything the plant needs. And one of those things it needs is glucose. And it'll take that glucose and convert it to different forms. It'll convert that glucose, plants will, into sucrose or fructose or galactose or any of these other stored sugars and such, and even into starch. It'll store it as starch. Plants store excess glucose as starch. You, being a heterotrophic, then you eat plants, or you eat things that eat plants, and then you'll bring that glucose in, and then you'll, or whatever, the starch, the glucose, the fat, the whatever, and you will break that down through a series of reactions to get it down to these monosaccharides, glucose, fructose, galactose, and then break that down and then move it into the cells. And then the cells will take that organic carbon, break the bonds, and in the process of breaking these bonds, convert the energy that was in those bonds into phosphate, a phosphate bond, into the ATP molecule. It's that ATP molecule then that moves around and drops off phosphates. When it drops off a phosphate, that is known as phosphorylation. It's moving energy from one form to another. You got to follow those thermodynamic laws. Life does it. So, like I mentioned, you get that, and you'll get in that glucose in there. So the glucose, or whatever the carbohydrate, whatever it is, you bring it in. And there's a series of steps: glycolysis, the transition reaction, Krebs cycle, electron transport chain. Those all require oxygen. Glycolysis doesn't. If you don't have any oxygen, then the glycolysis takes place, and then it'll go into what's known as a fermentation, where the end products in animal cells is usually lactic acid. In certain plants, certain yeast, certain bacteria, instead of lactic acid, it'll further break that down into alcohol and other things. So, but you can see right there, the overall reaction is the glucose plus the oxygen reacts to form carbon dioxide, water, and the energy. That's what we're focused on, the energy. We take that energy and we store it, or we don't store it, we move it to transform it to an ATP molecule and such. It's a pathway so that we get as much energy out as possible. We don't get it all out. Nature says you can't. The laws of thermodynamics say you can't. It's never 100%. In this process, it is pretty efficient when we do it aerobically. We'll take that glucose and we'll harvest about 38% of the energy that was there and such. Pretty good. In this process of breaking these bonds, these carbon to carbon bonds, these carbon to hydrogen bonds, these carbon to oxygen bonds and such, when you break it, the energy is in the electrons, if you will. And so we have these electron carriers, NADH and FADH2. They have a name to them and such. You can look it up. I might have it later on. I don't remember, because I don't try to memorize these names, because, golly, they're so big. But they pick up the electrons, and they move the electrons to the electron transport chain. That's their job. You have to have these NADH and FADH2. These are vitamins, if you think about it. I think they're part of your B vitamins. If there's no oxygen, they can't go to the mitochondria. So they have to turn on and drop the electron off somewhere. They usually drop it off on the closest organic molecule, which is the pyruvate, which is what's produced at the end of glycolysis. And then that breaks down into lactic acid. But if oxygen is present, they carry the electrons to the electron transport chain, and then that's a series of enzymes that move the electrons from one energy level to a lower energy level, and along the way stick a phosphate onto ATP. So, and from ADP to ATP. So let's start this thing. So we'll split the glucose. glycolysis. It is anaerobic. It is universal. It's been going on since life began. It's one of the oldest energy transformation reactions that we know of. So it happens whether oxygen is there or not. So we label it as an anaerobic respiration as such. Funny thing is, you got to put energy in to get energy out. So there's actually an endergonic portion to this where ATP has to be put in and such. But at the end of it, you actually make more ATP than you put in. So there's actually a yield of ATP in this one and such. Not big, but a small yield. And at the end of it, you get these two molecules known as pyruvate. This is an intermediate molecule, and what's the body going to do with the pyruvate? It depends on if oxygen is present or not. If it's present, that pyruvate then will be moved from the cytoplasm into the middle of the mitochondria. The middle of the mitochondria is known as the matrix. It's a solution with lots of enzymes and waters and other things necessary for cellular respiration. So the pyruvates move in. Pyruvate is a 3-carbon molecule, and it doesn't enter in as 3 carbons. So along the way from the glycolysis reactions to the Krebs cycle, it loses a carbon. That's known as decarboxylation. And it ends up with a 2-carbon compound, 2 carbons, and that's known as acetyl. That's an acetyl molecule. That gets picked up by this coenzyme, and it was one of the first ones discovered, so they call it coenzyme A. It forms this molecule, this is called acetyl-CoA, which then goes into the Krebs cycle and drops off the acetyl. The coenzyme A is like a Boy Scout, picks up another old lady to help her across the road. That's what it's doing. That acetyl, which is 2 carbons, combines with OAA, oxyl acetic acetate, if you will, and that forms a 6-carbon molecule. That 6-carbon molecule, which is stable, is known as citric acid. That then goes through a series of reactions where a carbon is stripped off, known as a decarboxylation. You get a 5-carbon molecule. Then it happens again, another decarboxylation. That's a 4-carbon molecule. That's succinyl acetate or something at that point. And then that is rearranged back into the original, the beginning 4-carbon molecule, OAA, oxalo acetate or oxaloacetic acid. So every time you strip off the carbon, you break a bond. NADH is there to pick it up. And then when we go through this rearranging, we break bonds. NADH is there to pick it up. FADH2 is also there to pick it up. And they grab these electrons and they move these electrons from the Krebs cycle to the electron transport system. So in the Krebs cycle, there's a couple of instances. Really, every time you put an acetyl in, you spin the cycle, you make 1 ATP. So there's a little bit of energy gain from this. The NADH, the FADH2, they take those harvested electrons and they move from the middle to the inner membrane known as the cristae, and they drop it off. Located on that cristae is a series of enzymes known as the electron transport chain or the electron transport system. They pick up the electron at one energy state and through a series of reactions, kind of like a slinky going down the stairs from one energy level to the next lower energy level to the next lower energy level. Hydrogen, which follows the electron carriers, is then pumped from the matrix into the inner membrane space. So you create a high concentration of hydrogen ions behind this membrane. They then flow through a special protein known as an ATP synthase. A hydrogen ion and a proton are the same. That'll confuse a lot of people because they switch the terms real fast in this stuff. So a hydrogen ion is also a proton. So in this inner membrane space, which is an area of the mitochondria between the outer membrane and the inner membrane, you have this gradient, this high concentration of hydrogen ions. And they flow from high concentration to low concentration. That's called osmosis. Remember that one? And since it's a chemical, they call it chemiosmosis. And as it flows down through this special protein known as an ATP synthase, it's an enzyme that synthesizes ATP. That's how ATP is produced in the mitochondria. And they have figured out that for each NADH, it has enough energy to make 3 ATP. And for each FADH2, it has enough energy to make 2 ATPs. So if you did the calculations and if you looked at it, for each molecule of glucose, you can get or harvest 36 ATP. That's the net. You actually made 38. It took 2 to start this thing, so you netted and such 36. There's some math in there. You can put energy values to these ATPs, and you can put energy values to what the original glucose was, and you can see that it's not 100%. It's actually just 38% of the energy available, which is really good and such. So, all right, we're going to stop right there.